On the study of the effect of protein on yeast mating pathway. My name is Vianney Ramirez. I am a sophomore at the Department of Biological Sciences at North Carolina State University. Outline. I will be going over the introduction, proposal, current research, methods, outcomes and challenges, and finishing off with the conclusion. Introduction. Saccharomyces cerevisiae, which is the yeast that I will be focusing on for this study, is commonly referred to as brewer's yeast. Brewer's yeast is used for fermented beverages and bread, processing food waste, and industrial ethanol production. It is easily cultured, making it a favorite for many researchers to use in labs. As many of us know, unicellular fungi reproduce through mitosis, and Saccharomyces cerevisiae participates in this process, but can also be a participant of meiosis. Proposal. The purpose of the proposal is to request a study on the evolutionary perspective of Saccharomyces cerevisiae mating type. The objectives and timeline include a study that will take 4 months to complete, including room for replication. So after the first 2 months, another study will be replicated the following period after that. The objective is to test the impact of the F-box protein on the mating signaling pathway. My hypothesis is that the F-box protein will increase the efficacy of yeast signaling transduction for reproduction. Ideally, if we look at the image beside us, we can see that all cultures have a difference of vitality, and the F-box protein would have more activity within the culture. Proposal continued. The experimental design focusing on growth conditions. 2 main Saccharomyces cerevisiae strains are used. We will be using BY4741, a genotype MATA Leu2met15, and BY4741 derived gene deletion mutants. These are exceptionally good for genetic manipulation because of their sensitivity to modifications. As I mentioned before, yeast, especially brewer's yeast, is very popular for genetic modification studies, which is why we're studying the mating pathway. Plasmid is essential for PCR and the expression of a GPA1 protein. Colonies will be grown overnight in a 30 Celsius environment. Colonies are subject to low nitrogen conditions by being suspended into ammonium sulfate lacking mediums. Once cultures are saturated, they will be diluted with a fresh medium. Autophagy means self-eating. At a subcellular level, in this experiment, it is the process in which cytoplasm is delivered into the yeast vacuole and degraded. Assays are used to monitor autophagy progression in cell cultures. For this experiment, PHO860 autophagy assay is performed similar to the previous study and research that was done prior to the proposal. A second assay is performed, the Rosella assay. I do want to highlight that it monitors the mating pheromone on the cultures that were deprived of nitrogen. A third assay is performed next. Morphogenesis assay measures the concentration of pheromone in the mating projections. So one of them is monitoring it, and the other one is measuring it. The transcription reporter assay is the next one, which quantifies the activity of proteins and their cell's growth. So they're all pretty much used for data, which we'll be collecting throughout the study. The budget plan that we are requesting is $10,000. 2/5 of it will be used for research assistance, such as research assistants, lab maintenance, materials. Yeast is fairly cheap, but microscopes do run on a more higher end of the cost. And we also want to have money left over to replicate the experiment. Current research, which we referred to earlier in the presentation. F-box protein is our main focus during this. So the research on evolution of yeast mating pathway has pointed out the influence of specific proteins on yeast mating signaling. And the F-box protein, being one of them, reports show that the mating pheromone factor promotes the yeast vacuolar targeting of a cytoplasmic reporter protein. Once the pheromone factor binds to its receptor, dissociation occurs in G protein in its subunits. So the research found aids in the scientific understanding of different methods in which yeast replicates itself under different conditions. And the better you understand the subject you're working with, the better you understand the research you're doing on it, such as any genetic modification that you are doing on it. That's why it's so important. Methods. The experimental design includes growth conditions, and this is just something that we went over already. I wanted to add images next to this to get a more in-detailed feeling of what the environment would look like and what types of materials would be used. Outcomes and challenges. This research will broaden our understanding of how pheromones from the F-box protein stimulate vacuolar targeting, leading to yeast production. The consequences of the GPA1 protein have not been studied in these experiments. When I say these experiments, I want to refer to not only my experiment, but previous research that I used for this. They have not touched base on any consequences that the GPA protein has on the yeast mating pathway. And the hypothesis created after the results determined the possibility that PS34 might also aid in the facilitation of the yeast mating pathway. So that is something I would be interested in researching later on after these studies are completed. New forms of the mating apparatus are supported through these results of the tested proteins in this experiment. There are many limitations that persist though, and I hope to close the gap on that in the future. Thank you for listening to my presentation. I will close it off with references.